In this video, we will discuss about a paper that talks about in vivo CRISPR knockout optimization on tumor restricted transgene expression. So what does this mean? This means that they want to optimize the in vivo CRISPR knockout system in a way that it will expressed in the tumor restricted region. So how they did that? So they first find out a promoter that is expressed more whenever the CAR T cells or the T cells interact with the tumors. So the optimizations are done in the T cells. So this is in the panel A, we can see their plan. So they took the target gene, which is they considered as payload, and put them in front of the tumor restricted promoter. So that is the promoter that is expressed more in the T cells when they are activated, and when the T cells, in which conditions T cells are activated when they interact with the tumors. So this is the tumor restricted promoter. So one of the promoters that they find out is this PDC D1 gene, or PDC D1 promoters. So they, in order to check their hypothesis whether this system will work, the payload or the target gene that they used first is the GFP. So this is, and they did it, and they checked and compare it with the wild type target gene that does not have the tumor restricted promoter, and the engineered target gene that has tumor restricted promoter, that has this payload just downstream of the tumor restricted promoters. And in the case of when this target gene, wild type target gene, engineered target gene face the tumors, then the payload with the engineered gene will express more. So in the panel B, we can see that they took the GFP protein and introduced this GFP protein just in front of this PDC P1, just in front of this PDC P1 promoter, or we can say that just in front of the start codon of this PDC P1 promoter that can be seen in the panel B. So now what they did is they first did the in vitro studies where they activate these T cells, and when the T cells are not activated, they are not able to see any high GFP expressions that can be seen in the first row. But when these cells are activated, which is indicated by high PD1 expression, and also we can see that high GFP expressions are also there, indicating that these uh, activated T cells, their principle that activated T cells has this high PDC-D1 expressions that will cause high GFP expressions. It seems to be working. Then they check it in the different cell lines. In all the cell lines, they are able to see high expressions of this GFP after 22 hours. Within 28 to 72 hours, they see a significant increase in the GFP signals, and compared to the non-stimulated case where the GFP signals are very low. Thereafter, once validating their results in the in vitro system, they want to check the specificity in the in vivo system. So for that, they took the system ovarian cancer cells and put them into the mice, and the mice will undergo treatment after 8 days of the tumor injections, and after 8 days of this treatment, they will analyze the CFP signals. And what they are able to see that in the case of spleen, where they do not expect any tumors, the GFP signals are low, and also these PD1 signals are low because tumors are not there in the spleen. But in the case where tumors are present, they are able to see the high PD1 expressions, indicating that more activated T cells are there, and the high GFP expressions are also there. So this indicates that their specificity is also good, that the expressions of their target genes are occurs at the site of tumors, and these things are quantified in the right side of the panel F, where we can see that GFP expressions are high in the tumors but low in the spleen. Once validating their system with GFP, they wanted to check it whether it can work for actual transgenes that are important to tackle with the tumors. So in this case, they use different cytokines. First, they use it with TNF, and they can see that when they use PD1 TNF, the tumors areas are in the lower side, and similarly for other cytokines such as IFN gamma, interleukin 12, they are able to see when these are used, the tumors area is lower, indicating that they are able to perform its functions that is expected to reduce the tumor formations or slow down the growth of tumors. And even for this IFN alpha 9 also they are able to see a IFN 9 alpha and IFN beta, they are able to see a decreased tumors area. However, for the interleukin 12, when they check, although for interleukin 12 when they check, they are able to see systematic toxicity, that the cells are that the mice die after a few days, and the systematic toxicities leads to a side effect that cause this decrease in the weight of the mice when they use IR12. And the reason that they think is happened because of the high expressions of this transgene, that is the PDC7 at the periphery of the tumors, and that will cause the high expressions of the interleukin 2, and that leads to the side effect in terms of the high systematic toxicity. So they finally conclude that they need to find out a different transgene expressions that is not overexpressed uh, in such a way that is not overexpressed or above a threshold in such a way that will cause the systematic toxicities. So this is what they concluded from their first figure, or the experiments that are shown in the first figure. I hope you understood how they are checking their in vivo CRISPR knockout system on and make them the tumor restricted, make them specific to the tumor restricted transgene expressions. In this case, they have checked both PDC given, but they have also find out one problems that it will cause systematic toxicities. We will discuss in subsequent videos how they tackle up these problems and what are the approaches they used to optimize their in vivo CRISPR knockout that is specific to the tumor restricted transgene expressions. That's all for this video. If you need to understand more about the experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.